Hello, Assalamu alaikum, dear students. You are welcome again on online class of fermentation and its industrial applications. Lecture number 23, and we were talking on fermented foods. So we are going in detail to talk on fermented foods with continuation of our previous lecture. So this is lecture number 23 in fermented foods. In our previous class, we talked about what are the fermented foods, and here we are talking about the cassava. What is cassava? It is a fermented food. Fresh cassava contains anhydric acid that should be eliminated from any product originating from cassava to render it fit for human consumption, depending on the production method, particularly traditional method. Gari could contain up to 20 milligram per kg of anhydrin against 43 milligram per kg for fresh peeled cassava. Gari is a fermented, gelled, and dehydrated food produced from fresh cassava. It is a popular diet in Nigeria, Benin, Togo, Ghana, and in other West African countries. The consumption area even expands to Central Africa, Gabon, Cameroon, Congo, Brazil, and Angola. Polvillo is a fine tapioca Monioso cassava flour. It can be found at Latino markets in California as sour starch or sweet starch. So, Nata de Coco, a higher fiber, zero fat Filipino dessert. It is also food type. A chewy translucent jelly-like food product produced by the bacterial fermentation of coconut milk. Commonly sweetened as a candy or dessert, and can accompany many things, including pickles, drinks, ice cream, and fruit mixes. Highly regarded for its high dietary fiber and it is zero fat and cholesterol content. But we should remember that it is produced by fermentation of coconut milk. It is produced through a series of steps ranging from milk extraction from coconut, mixing fermentation, separation, cleaning, cutting to packaging. There are various steps are involved and all are related and all are a, a total fermentation process in which you are taking raw material, you are extracting milk from raw material, mixing it with some substances, fermenting it, separating it, cleaning it, cutting it, and packing it. Then we are sending it to the market. So a huge industry can be covered through this product. And for this fermentation, mostly we are using lactic acid bacteria, a major group of the fermentative organisms. For fermentation of the raw foods to convert into desirable food substances, we are using lactic acid bacteria. It is a major group of the fermentative microorganisms. This group is comprised of 11 genera of gram-positive bacteria, If you will see the names here, Corynebacterium, Onychococcus, Enterococcus, Pediococcus, Lactococcus, Streptococcus, Lactobacillus, Vagococcus, Lactosphaera, Vessels, and Lichenostock. These are the bacteria which are used for the fermentation of the product. Why? Why these bacteria are used? Because they are producing lactic acid. In presence of acid, the nature of the raw material changes into another flavor. Related to this group are genera such as Aerococcus, Mycobacterium, and Propionibacterium are also used for the fermentation of foods. So, from every species have characteristic that they have ability to produce unique taste and flavor from in every carbohydrate containing raw foods. When microorganisms and raw food are incubated for fermentation, that every species have capability to produce unique type of flavor and aroma from raw material or from raw food to fermented foods. Lactic acid bacteria, while this is a loosely defined group with no precise boundaries, it has no clear-cut boundaries. All members share the property of producing lactic acid from exhaust. So by using sugar, these bacteria have ability to produce acid. Most of the members of this, they have ability to produce lactic acid by using sugars. As fermenting organisms, they lack functional heme-linked electron transport system or cytochromes. They don't have a functional Krebs cycle. Energy is obtained by substrate level phosphorylation while oxidizing carbohydrates. They are getting energy from this, producing lactic acid, and that acid changes the nature of the food and gives a specific aroma and flavor to the food by by breaking down the food's basic content, basic sugars of the food which are present over there. The lactic acid bacteria can be divided into two groups based on the end product of glucose metabolism. Those bacteria that produce lactic acid as the major are sole product of glucose fermentation are designated homofermented. Those bacteria that produce lactic acid as the major or sole product of glucose fermentation are designated homofermented. Those bacteria that produce equal amount of lactic acid, ethanol, and carbon dioxide are called heterofermented bacteria. The homolactics are able to extract about twice as much energy from a given quantity of glucose as the heterolactics. Means homolactics extract more energy from any quantity of sugar compared to heterolactic acid producing bacteria. All members of Pedococcus, Lactococcus, Streptococcus, Vagococcus, along with some lactobacilli are homofermented. Corynebacterium, Oinococcus, Enterococcus, Lactosphaera, and Vessels and Lichenostock and some lactobacilli are heterofermenters. The heterolactics are more important than the homolactics in producing flavor and aroma components such as acetaldehyde and diacetyls. Because heterofermenters can ferment variety of the foodstuffs and can produce variety of the aroma and flavors from variety of the food substances. Therefore, heterofermenters are considered very important and very good in fermentation industry of food. Lactic acid bacteria growth. The lactic acid bacteria are mesophiles, and you know the mesophilic. They generally grow our temperature range of about 10 to 40 degrees C, and optimum between 25 to 35 degrees C. They have good growth. Some can grow below 5 and as high as 45. So you can consider these are the extremophiles which can grow at below 5 degrees C as well as above 45 degrees C. Most can grow in the pH range from 4 to 8, some as low as 3.2 as high, and some in high pH as 9.6. So these are some extremophiles are also present over here, which can grow at extreme conditions to protect and can ferment the food at that pH. So starter culture, when we are preparing the raw material of food to ferment and to convert it into our desired food and desired product, either in domestic scale, in pilot scale, or in industrial scale. But for that, we are growing microorganisms in laboratory or in industry to use as a starter culture, because before adding starter culture of the specific microorganism uh, to produce specific aroma and flavor in special food, in specific food, because we have to clear one thing over here. Every microorganism have ability to produce unique type of aroma and flavor from one substrate. So here we are compelled, here it is compulsion on us that we have to make the starter culture, pure starter culture of that particular microorganism which we want to use for fermentation. Traditionally, the fermenting organism came from the natural microflora or a portion of the previous fermentation. Okay. Traditionally, it came from where? It came from nature, from a wild environment, or it came from previous fermentation, such as we are taking a small amount of yogurt from previous to add into the fresh milk to form new yogurt. The same principle is working, either that is going on industrial basis or on commercial basis, in pilot scale basis or domestic basis. In many cases, the natural microflora is either insufficient, uncontrollable, and unpredictable, or is destroyed during preparation of the sample prior to fermentation, e.g., pasteurization. So we are not preferring the wild microorganism, because when we are autoclaving and sterilizing the raw material to eliminate all other microorganisms which can damage that food material if you will ferment with those microorganisms in that food substance, we are sterilizing it. So all microorganisms which are present at that food material, those will be killed, those will be finished. So we have to add a new and a fresh microorganism which has been obtained, optimized, purified, identified in laboratory, that is called starter culture. A starter culture can provide particular characteristics in a more controlled and predictable fermentation, because about starter culture, we are sure that microorganism we have, this microorganism have this ability in short form. We have no doubt about this, that it will damage or it will destroy the food material over here. So starter culture Lactic acid starters always include bacteria that convert sugars to lactic acid usually. So for this we are using such microorganisms which have ability to convert sugars into lactic acid. Lactococcus lactis, subspecies lactis, Lactococcus lactis subspecies Ceremoras, Lactococcus lactis subspecies Lactis biovert diacetylactic, where flavor and aroma compounds such as diacetyl are desired that lactic acid starters will include heterofermentative organisms such as lichenostoc, citrovorum, and lichenostoc dextronicum. These species also have ability to produce aroma in uh, carbohydrate containing foods during fermentation. So, starter culture, the primary function of lactic acid starters is production of lactic acid from sugar. When we are adding a starter culture microorganism, they are using sugar and they are producing lactic acid from this sugar. Other functions of starter culture may include the following: that starter culture of microorganisms gives flavor, aroma, and alcohol production. Flavor attracts customers, aroma also attracts customers, and alcohol which preserves the food for longer time. Proteolytic and lipolytic activities, they are also breaking down the proteins and lipids, inhibition of the undesirable organisms, and they are restricting the addition or entrance of other microorganisms during this by changing its pH. A good starter culture will convert most of the sugars to lactic acid that will use all material. Increase the lactic acid concentration to 0.8 to 1.2%. Titrable acidity. So draw this amount of lactic acid drops the pH between 4.3 to 4.5. So normal flora, the bacteria which were normally growing at neutral pH, now they are unable to grow our tears. Food scientists frequently use the ability of bacterial cells to grow and form colonies on solid media to isolate bacteria from foods. To determine what type and how many bacteria are present, we are using strict flat technique. First, we are taking food scientists frequently use the ability of bacterial cells to grow and form colonies on solid media to isolate bacteria from foods. First, we are isolating bacteria from food to determine that which type of microorganism is here, and we are characterizing that microorganism, that this microorganism have ability to produce which type of aroma, which type of flavor, and which type of organic acid it is producing, and how they can, for how longer it can make attractive food for the consumers, and how long it preserves the food for longer time. So we are using a strict plate technique to isolate bacteria from food material. If that is uniform culture, okay, this all organism is one species. Further, we are characterizing it. The streak plate technique, bacteria are streaked over the surface of an agar plate so as to obtain single colony. Obtaining single colonies is important as it enables the size of bacteria, shape of bacteria, and color of the individual colony to be examined. It can also highlight the presence of contaminating microorganisms. So we are using isolated colonies. We are trying to obtain isolated colonies on nutrient agar plate. If all colonies are similar, they are producing same pigment. Colonial morphology is similar, pigmentation is similar, colonial size is similar. So we are assuming that only one type of microbial colony is present in food material. If there are various type of colonies, so we are thinking that it is contaminated. So if you will add such type of starter culture for fermentation, it can destroy, it can damage the food material, instead of fermentation to convert it into a attractive and fermented food. So we have to be so microbiology person now and can tackle this all situation. You must have to monitor during this during addition of the starter culture. You must have to identify and make sure that there is no contamination in starter culture, and only your desired microorganism is our dear. So streak plate technique, using a loop to streak plate, a loop. So you can use such like of, you can obtain, you just you will pick one colony from food material and will dip into the food and will streak it and make it like this. This is the technique. And after incubation, you will find different colonies over here, and every colony has been produced from one bacterial cell. When conditions are right, two bacteria can double in number every 20 minutes. When condition, because we are fermenting, in fermentation we are keeping condition suitable for the growth of microorganisms. So microbial growth rapidly increases. They are going in log phase and they are converting all available food material into lactic acid, aroma, and flavor. And uh, after completion of fermentation, you can preserve it. So for the isolation of microorganisms, microscopic examination, by microscopic examination, we can identify the microorganism. It can provide information on to the what is the size of the bacteria, what is the shape of bacteria, either these are bacillus, either these are cocci, either these are spiral, what type of this rods, cocci, and spiral. It cannot provide enough information to enable bacteria to be identified. Microscopy give a limited information uh, about the microorganism. But for complete characterization, that which type of metabolites it can use, it can produce, which type of aroma from which material it can produce, so we have to go in detailed study. This is the microscopic view of stained bacteria. We are coloring sometime microorganism to see this thing. So these were some examples of some examples of the fermented food, which are in practice throughout the globe. Either this is going on traditional and small scale, either these are going in industrial scale. If further examples, if you want to see, you have many examples. All organic acids which are produced through fermentation by microorganism, uh, such as alcohol, the citric acids, and many things. Beverages, all the items fermented beverages and alcohols are produced by uh, means of the microorganism, by using microorganisms. And another foods, breads, dough, and everything which is fermented are produced